Okay, last time we introduced the introduction part of medical microbiology. We saw that we have a big picture of this field, like the general history, what happened in history, the big guys, the famous scientists who contributed a lot in this field, and we also know about the important part in our course, and we mentioned that we'll teach bacteriology and virology a lot in the following 8 weeks. So today we'll begin learning bacteriology. So all about bacteria. So we'll spend maybe 6 weeks on bacteria. So for the first time, we need to know the morphology and structure of bacteria. Morphology means, anyone know the meaning of morphology? Morphology is like the shape, how they look like. And structure, we need to dissect the cell so that we know how the bacterial cells are made up, what important structure, what important parts in the cell. So that's what we are going to learn today. Okay, so first we want to know, what are bacteria? Definition of bacteria here, they are unicellular prokaryotic organisms that have no organelles except ribosomes, and they don't have nuclear enclosed, membrane enclosed nucleus. So the key points here, first, prokaryotic. So that's the term we introduced last time. Prokaryotic means they don't have real nucleus. So that's the same meaning as the last sentence, last term. And also unicellular. The meaning of unicellular means, so they are single cells. Yeah. So unicorn, right? Unicellular. But even though they are single cell organisms, it doesn't mean that all bacteria live only one by one. Sometimes they need to form a big group so that they can live together. So that's the organization or grouping we'll talk about. And they are relatively small, smaller than eukaryotic cells. So bacteria are relatively small, and also relatively simple structure. Single cell, simple structure. So that's what we'll focus on, the structure today. Size of bacteria. Most of the bacteria are on the size of micron or micrometer. We introduced that last time. 1 micrometer is 10 to minus 3 millimeters. So that's micrometer, very small. And we need to rely on either light microscope or electron microscope to study bacteria. But that's the general size. When we talk about biology, we should keep in mind that it's high diversity, and there are always exceptions in this field. And when we say that most bacteria are on the size of micrometer, it doesn't mean the bacteria can be longer or smaller. So some bacteria are really big, and you can actually see it with your eyes. Let me give you some example later. And some bacteria are very small. They are a little bit bigger than viruses, and they can pass through the filter membrane. The filter membrane, usually we use it to block bacteria, to remove bacteria from solution. But some bacteria can actually pass through the membrane because they are very small. Okay, here are the major shapes of bacteria. Most of the bacteria look like this. Either, okay, some of them are coccus, Do you know the meaning of coccus? Coccus is a name in Greek we call berry, like blueberry, the berry-like coccus. So coccus is this spherical structure, like a ball, a sphere. So that's coccus. And so the diameter of the coccus is usually around 1 micrometer, so very small. And when you say lot of coccus, you can use the term cocci. So that means more than one. Another shape is rod-like this one, rod-like. It's like this charcoal. Sorry, what's this? I cannot remember the name. I learned this in primary school. It's like this pen, like a rod. And this type of bacteria, we call them bacillus. So this is the Greek name, bacillus. And for more than one, it's bacilli. And these rods, usually they are less than 1 micrometer wide, less than 1 micrometer wide. And the length varies. Some can be 3 micrometer or 1 to 3 micrometer. The big ones, like anthrax, we mentioned that last time, the first bacteria ever identified, which can be 3 to 10 micrometer long, so it's very long. And another group of bacteria, we call them spiral bacteria. They look like this. Okay, different type of spiral bacteria. Okay, and one of them is vibrio. We'll talk about that when we learn the specific pathogens. So these are the major shapes of bacteria. But keep in mind of diversity. Some bacteria might look quite weird to you. They are not looking like this, but they look weird. But that's normal for biology. Okay, like this one. This is a newly covered bacteria. The name of this one is Magnifica thiomagrata. So it's a bacteria recently discovered, and it was published in Science in 2022. And this bacteria can be very long, can be like as long as like centimeter long, 1 to 2 centimeter long. So this long, so that you can actually see it, and it might carry multiple copies of their DNA. But for some reason, they fail to divide into small cells so that they can be very long to form this thread-like structure. This is a newly covered bacteria. And yeah, of course, this one, we don't have it in our final exam, just to give you some information, okay? And grouping of bacteria. And even though we call them unicellular and they are single cell organisms, but it doesn't mean that they prefer to live one by one, like a single cell floating in the air or a single cell floating in the water, because that's not good for the bacteria, for the cell. It's hard for them to defend themselves, to keep themselves safe. So usually the bacteria like to group together. So a lot of cells come together, bind to each other, and this binding, connecting to each other, we call them grouping of bacteria. Some, they form pairs, like this one. So bacteria in a pair means we call them diplococci. Do you know the meaning of diplo? Di means two. So diplococci, two bacteria to form a pair. Or a lot of bacteria can form a chain, like this one. It's a long chain. So if it's a chain, we call them strap. Strap means a chain. So this one we call them streptococcus. Or some bacteria can form cubical bundle, like 4 bacteria here to form a 4-cell bundle. So it's a tetrad. Or this one, 8 cells to bond together. We call them succina. Succina means 8, right? Or some bacteria, they group together like irregular cluster. Irregular cluster means like you have a bunch of grapes. So the grapes bounce together irregularly. So this one we call them grape-like irregular cluster. The famous example of this one is Staphylococcus. In this name, the first part, Staphy, means grape. So Staphylococcus. And one of our major pathogens we'll introduce in detail. And that's for coccus. For bacillus, they can be like, some of them prefer to live in single cell. So it can be single, or they can be in pair, or some of them like to form a long chain. Okay, so this one we call streptobacilli. Strepto also means chain, so streptobacilli. And some bacteria, they like to, well, lay next to each other in parallel, like this one. The one we will introduce as Corynebacterium diphtheria. So they lay in parallel to next to each other. So that's the bacilli. Okay, so this is a major important for today. We want to introduce the structure and function of bacterial cells. So keep in mind that when we learn something, when we introduce something, we always want to introduce their structure, so that's the material, together with their function. And we know the function relies on the structure. Why it look like this? Because it has to carry out some specific function. So we introduce structure and function at the same time. Okay, so this is the model structure of bacterial cell with bacillus as an example. And of course, it's not a real cell, but with this model, we dissect the cell and we can find several essential structures. Essential structures means that these structures are common structure. It appears in all bacterial cells. That includes, well, cell wall. So you can find cell wall in almost all type of bacteria, but I mean not all. Almost all. Some bacteria actually don't have cell wall. We'll introduce that later. And also they have cell membrane, the periplasmic membrane. And also inside the cell, that's a cytoplasm. And they have their nuclear material. Even though they don't have a nucleus, but they do have their DNA, the nuclear material, which condensed in part of the cell. It's not commonly well distributed, or evenly distributed in the cell, but it's condensed in part of the cell, inside the cell. So that's the essential structures. It can be found in almost all bacteria. And we also introduce some particular bacteria structures. The particular structures that include the capsule, which is an additional layer of material surrounding the cell. Outside the cell, there is a special component surrounding the cell. And also, some bacteria has flagella, which is the locomotion organ of the cell. This flagella can move so that it can propel the cell to swim or to move. And also pili, so that's this hair-like structure. That's a pili on the cell surface. And some bacteria can form spores. Not all bacteria can form spores. Some bacteria, especially some gram-positive bacteria, can form spores, and which the spores are very resistant to harsh conditions, so that the spores can survive in like very dry condition or very high temperature. I will introduce these contents one by one in detail. Okay, the first one is the cell wall. The cell wall is some rigid material outside the membrane. You know, the membrane, when we learn in high school, we know that the membrane is kind of mosaic fluid, right? So we know the mosaic fluid model. So it can flow. So it's not rigid, it's not hard. But outside the membrane, here is the membrane, thin layer of membrane. You can find a layer of material which is hard, which is rigid, like the brown layer. This is the gram-positive bacteria. And this layer is peptide glycan. We'll introduce the structure on the molecular level of peptide glycan. Okay? So it's kind of hard. You cannot push it. So that keeps the specific shape of the bacterial cell. Without the cell wall, it cannot keep the specific shape. So this is gram-positive cell wall. For gram-negative, this is the membrane, the pink one, and the peptide glycan layer is here. It's very thin, only a couple of layers of peptide glycan. But outside the peptide glycan, you will find another layer we call outer membrane, the outer membrane. So in gram-negative bacteria, there are two layers of membrane. One is the periplasmic membrane, the other one is outer membrane. And for the cell wall, when we say cell wall, it means the outer membrane and the peptide glycan. So the two layers together to form the cell wall of gram-negative bacteria. So this is the structure under electron microscope. You can actually see the structure, cell wall structure, under electron microscope. But there's one thing I need to tell you. The color is not real. Okay? So this brown color or pink color, we call pseudo color. After we take the picture, we give them label them with some specific color. But under electron microscope, it's all black and white, no color at all. Okay? Same here. Okay, the cell wall. The major components of the cell wall is peptide glycan. It contains the backbone. The backbone is composed of alternating saccharides. One is N-acetyl glucosamine, and the other one is acetyl muramic acid. It's two sugar molecules. You don't have to know the exact structure of these two sugar molecules. You can imagine that two sugar molecules to link together head to tail by beta 1,4 glycosidic bond. Let me show you the structure later. So that's the backbone. And they also have the tetrapeptide side chain. So that's the side chain on the backbone. And also they have the pentapeptide cross bridge. Do you know the meaning of pent? Five, right? Five. So the pentapeptide cross bridge means that it's a peptide with 5 amino acids. So 5 amino acids. Let me show you the structure. So here is the peptide glycan layer of gram-positive bacteria. First, the backbone here. The G means acetyl glucosamine, and M means N-acetyl muramic acid. So here GM, 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 GM. So the alternating structure. Okay, to link together, the bond between the two saccharides, two sugar molecule is 1,4 beta glycosidic bond. Okay, it's here 1,4 glycosidic bond. So that's a backbone. And on the backbone linked to M molecule, the muramic acid, you can see the 4 peptide side chain. So 4 amino acids side chain on M. All are on M. Okay? So that's a side chain. And we have the cross bridge, 5 amino acid cross bridge. So that's the purple one. 1, 2, 3, 4, 5. Okay? All of them are glycine amino acid. So 5 glycine, okay, to link the alanine and the lysine, okay, to link together. So this is a one layer of peptidoglycan in gram-positive bacteria. Just one layer. And even on this one layer, the 2D structure, you can sense that it's very stable, right? So it has a backbone and a side chain and cross bridge links them together. So this is a stable structure. And in the actual cell wall, it can be very thick, like this. It's because it contains a lot of layers of this peptidoglycan. Sometimes it can be over 80 layers of peptidoglycan in the cell wall. So which makes the cell wall very hard, very rigid, so that it's not easy to break them. I used to work on gram-negative bacteria for a long time. In recent years, I worked on gram-positive bacteria. I want to lyse a staphylococcus, but I feel that it's so hard in the lab to lyse the cell, to break the cell because of the cell wall. We should usually we treat the cell with enzyme to digest the cell wall in the lab for over half a day so that we can finally break the cell wall. So it's very, very rigid, very hard to break it. So yeah, this is a gram-positive bacterial cell wall, the peptidoglycan. Okay. For gram-negative bacteria, it's quite different. Yeah, you stop talking. Sorry, guys. Gram-negative bacteria. Okay, here it's the same. The backbone is the same. Here, GM, alternating saccharide. Okay, and they are linked through the beta 1,4 glycosidic bond. Okay, but here the side chain, even though it's on M, okay, but you cannot see the glycine cross bridge. See, between the two side chains, you cannot see the 5 glycine cross bridge, like this one, right? So in gram-negative bacteria, you can see the structure is kind of loose, right? It's not stable, it's not tightly packed together. So that the peptidoglycan in gram-negative bacteria is very weak. So with some mechanic methods, you can just break it so that you can lyse the cell easily. So this is gram-negative bacteria. But sometimes in the gram-negative bacteria, the side chain, even though there is no glycine cross bridge, sometimes they can link together directly. Okay? Occasionally, they can link together directly, but not in every side chain. So that this structure is kind of loose and you can break it easier than gram-positive bacterial peptide glycan. So that's the major component in the cell wall of gram-positive and gram-negative bacteria. Okay. A special component of gram-positive cell wall is the picoic acid. This is how to read this word. Picoic acid. Picoic acid is also a polymer, okay? which can be covalently linked to either the membrane. Okay, this is a membrane. It's covalently linked to the lipid molecule, or it can be covalently linked to peptidoglycan, like this one. It's linked to the backbone. If it's linked to the membrane, we call them membrane teichoic acid. Okay. If it's linked to the cell wall, okay, we call it wall teichoic acid. It's covalently linked to the peptidoglycan. Again, it's a mirapping acid. And this is a special component of gram-positive bacterial cell wall. And the structure of the teichoic acid, like their charge and their modification, is very important for bacterial antimicrobial resistance. And yeah, luckily in recent couple of years, I had one master student. We worked on the teichoic acid modification, and we had one paper published. So if some modification on this molecule changed, the bacteria can be more resistant to chemicals, to medicine. So this is the special component of the gram-positive bacteria. And also the teichoic acid extends through the cell wall, and it's the major surface antigen of the cell. That's not hard to understand because they extend out of the cell, so it's exposed to the immune system, so that your immune system can react to these components to make either antibody or that they can stimulate immune cells like T cells. So that's the surface antigens. Okay, this is gram-positive. Okay, you don't have to remember this one, just to show you how they look like, okay? And for gram-negative bacteria, the special component of the gram-negative cell wall is the outer membrane, okay? This is the periplasmic membrane, okay? And this is the peptidoglycan. You can see only a couple of layers, very thin. And on the outside of the peptidoglycan layer here, you will see another layer of membrane. We call it outer membrane. And outer membrane together with the peptidoglycan, they make the cell wall. And on the outer membrane, you can see that the outer leaflet and the inner leaflet is different. See here, the color. The inner leaflet and the outer leaflet is different. For the inner leaflet here, it's made up mainly by phospholipids, like this one, phospholipid. But on the outer part here, it's made up by LPS, lipopolysaccharide. Okay, lipopolysaccharide. Do you know the meaning of LPS? What's the meaning, lipo? Yeah, it contains lipid. Poly means multiple, a lot. Saccharide, sugar, right? So that's tell us the major structure, major components in LPS. Let me show you. But also on the outer membrane, you will find, well, lipid bilayer and also these proteins or lipoproteins. That's the proteins modified by addition of lipid molecules, or some of them are glycolipid proteins, some proteins which are modified by adding saccharide molecules. Let me show you the structure of LPS. LPS, okay, this is a general structure of E. coli LPS. First, the very outside of the molecule is this part. Okay, this part we call specific polysaccharide. Specific because it's specific to the species. One species will carry its own specific outer antigen. So that's the O antigen. When we say O antigen of some bacteria, we are saying about this part. So this is the O antigen. Okay, for E. coli, well, this species might have a specific O antigen. And for another cell belong to the same genus, they might have a different O antigen. So this is specific to the species. And then the middle part, it's polysaccharide. But this polysaccharide we call it core polysaccharide. It's not specific to the species. It's specific to the genus. A different species of bacteria belong to the same genus might share the core polysaccharide. Okay, so that's polysaccharide. And how about the lipo part? It's here. Okay, this part we call lipid A. Lipid A. And this is a hydrophobic part, right? Because it contains lipid, it's hydrophobic part. So it paired together to form the outer leaflet of the outer membrane, the lipid A. Lipid A is associated with the toxicity of LPS, and it has no species specificity. LPS from different gram-negative bacteria will carry out similar function. For example, they can induce our body temperature to raise. So all due to the presence of lipid A, not their O antigen or core antigen. Okay, lipid A carry out the toxicity of this molecule. Yeah, LPS is very important for either research or for clinical practice. When we give our patients some medicine, especially some liquid into the body, you want to make sure that it does not contain any LPS, or it can induce some problem based on the amount of LPS contaminated the liquid. So that's the LPS. And LPS is very stable because it does not contain any protein structure in it. It's just polysaccharide. So it's very stable. In the lab, if we want to make sure that all LPS molecules are destroyed, like in a glassware, we need to bake the glassware in the oven for over several hours to bake it. It's actually a baker. You put the glassware in it and raise the temperature over 250 degrees and keep it for 4 hours to destroy LPS. Very stable. Okay. Another structure we want to introduce is called periplasmic space. Periplasmic space. Do you understand the meaning of peri? What's the meaning of peri? Peri means surrounding, right? So surrounding. Plasmic means like the plasm. Periplasmic space is the space surrounding the membrane, the plasmic membrane. So traditionally, we only introduce the periplasmic space in gram-negative bacteria. So the gram-negative bacteria, the periplasmic space is a space between the two-layer membrane, the outer membrane and the plasmic membrane. So there is a gap, you can see, between this layer of membrane and this layer of membrane. There is a gap or space, very thin space. We call it periplasmic space. Okay. and you can see your peptide glycan in the space, okay? And that's for gram-negative bacteria. For gram-positive bacteria, for a long time, people don't know there is presence of periplasmic space. But in recent years, you can find some literature, some paper, they report the presence of periplasmic space in gram-positive bacteria. So this is a membrane, the green part, deep green part. And outside the membrane and inside the peptide glycan, you can see a space, a very narrow space. And that space we call it periplasmic space. So that's in gram-positive bacteria. Keep that in mind, just something new we want to learn. Okay? And yeah, we introduced the structure of the cell wall. What's their functions? Why the bacteria have the cell wall? How the cell wall contributes to the cell? The first one is that we mentioned that because of the presence of peptide glycan, and those material are kind of rigid, so that they can maintain the cell's characteristic shape. Like this species, it's a ball, it's a sphere, so that the wall keeps the sphere structure. While this one, it's a bacillus. The reason why it's bacillus is because the cell wall is in the shape of bacillus, so that it can maintain the cell's characteristic shape and giving the osmotic protection. You know the meaning of osmotic? Yeah, osmotic pressure is very important for life. The human cell in pure water, what will happen? The water will get inside the cell and the cell will swell and finally lyse and die, right? The reason it will swell and lyse is because there's no protection layer outside the cell, right? But for bacteria, because the presence of cell wall, even you put E. coli into pure water, the water can get in, but it's okay because the presence of cell wall, it does not swell and die. So that's the osmotic protection. So that's why for bacteria, they can live in different conditions, even in like the fresh water, like in the water in our lake. The osmotic pressure in the lake is low, and the bacteria can live there and replicate in the lake. That's because of presence of cell wall. And they play an essential role in cell division. And they should play some very important role in cell division, but probably we don't know exactly how it works. For example, you have a staphylococcus. It's a sphere, and it's surrounded by a thick layer of cell wall. And we know that the glycococcus, when they divide, they will form from one cell to two cells. So there must be something happening on the wall, right? You need to use some enzyme to break the original wall, separate the cell, and then to form two walls. So the cell wall, even though it's hard, it's rigid, it doesn't mean that it's like a frozen structure. It's changing all the time. And you need, when a cell grows from small to big, you need to break down the cell wall to form new walls. And when it's split from one cell to two cells, you need to split the cell wall also. So it's a very dynamic structure. And the cell wall is the site of major antigenic determinants of the cell, because the cell wall is on the very outside of the cell, and it can be detected by the immune system so that they play the antigenic role in the interplay between the bacteria and the immune system. Yeah, this one, the gram stain, we'll introduce a little bit. When you go to the lab, the second lab, I guess, you will do this experiment. And you will have a mixture of bacteria containing both gram-positive and gram-negative cell. And use this method, you stain the cell and observe it under the microscope, so that after staining under the microscope, you can tell which one is positive, which one is negative. Okay, so it's a very simple method, including 4 steps. The first time you stain the cell with crystal violet, which is a purple dye, it can bind to the cell wall and also get into the cell. And then after that, you stain it with iodine. Iodine is another molecule which can fix the dye to the cell wall so that it can be hard to be washed off. And the next step, we need to decolor the staining. Usually we use ethanol, okay? Ethanol to wash the cell. For the gram-negative bacteria, because of the outer membrane contains lipids, and the ethanol can break the lipids so that it can decolorize the gram-negative bacteria. But for the gram-positive bacteria, because the cell wall is so thick and so condensed, it cannot decolorize the color. After that, we counterstain with another red dye, a pink dye, a red dye, so that the gram-negative bacteria will be stained with pink color, while the gram-positive will stay in purple color. Okay, you will do this in the lab. We'll have a break. All right. Okay, just now we introduced the first important essential structure, the cell wall of bacterial cell. Sometimes the bacteria, when either they are treated with some enzyme or some medicine such as antibiotics, or they can be spontaneously changing themselves, like they get rid of their own cell wall. So that happens in patients, especially inside our body. And if a bacteria doesn't have a cell wall, they get rid of their cell wall. And those, we call them L-form bacteria, L-form. So it can be happen when the bacteria are treated with either enzyme or antibiotic. They just get rid of their own cell wall. So it's called L-form. And L-form, they don't have their specific structure, the characteristic shape anymore. So it's really hard for the doctors to diagnose the patient. Well, they know, well, it's some kind of infection. But what type of bacteria is really hard to say because they look quite weird. Let me show you. So this is a cell wall, the peptidoglycan. If you treat with like litholine, it's an enzyme which can break the backbone, the saccharide backbone, to cut it into pieces. Or if you treat it with penicillin, the first antibiotic, the acting site of penicillin is here on the cross bridge, so that both of them can remove the cell wall or inhibit the formation of cell wall. So it will form a cell without cell wall, we call L form. There is one interesting knowledge you want to know is that both litholine and penicillin were discovered by the same scientist, Dr. Alexander Fleming. He discovered both. So to me, the discovery of lysozyme also can give him one Nobel Prize. Okay. The features of L form. First, the L forms can be produced spontaneously or can be induced. And L forms are difficult to be cultured in the lab because without the protection of cell wall, you have to provide a special condition. For example, the osmotic pressure to culture the L form because there's no wall to protect them. And the other property is that the L form cell can be reverted to its normal form, which means that they can regain their cell wall to the normal state, revert to the normal form. And if it happens in the patient's body, in human body, the L form may cause some chronic infection, which is not easy to be cured. For example, if some person get infected with Staphylococcus, which is a gram-positive bacteria, you can give the person penicillin because penicillin can inhibit the production of cell wall, inhibit the formation of cell wall, so that you can finally kill the bacteria. But if it's already L form, it does not have a cell wall. So if you use antibiotics targeting on the cell wall, it does not work anymore because you lost your target. So it's not easy to be cultured, to be treated in the patient when they are infected with L form. This is how they look like. This is the normal form of Staphylococcus, and it's the grape-like cluster. It's easy to be identified, but that's the L form. So if in the hospital, from the specimen, you discovered a structure like this, it's really hard to tell what they are because they are not typical structure. Okay, cell form. But this form can be reverted to the normal form when the condition is proper. And when we culture the L form in the lab, we need to provide proper osmotic pressure so that they can grow slowly to form a special colonies. And those colonies, we say that they look like fried egg, like this one, the fried egg colonies. Okay. All right. The second important structure we want to introduce is the cell membrane. And we know the cell membrane is made up by what? The two layer of phospholipid, right? Phospholipid because it has this phosphate group which is hydrophilic, and it contains a tail part with what's the tail? Fatty acid, right? The fatty acid tail. So that the fatty acids tail are hydrophobic. So the cell membrane contains two layers of hydrophilic surface, and the inner part is hydrophobic. This makes the cell membrane, well, semi-permeable, so that big molecules cannot pass through the membrane. Only small molecules, especially those non-polar small molecules, can pass through the membrane. For example, oxygen. Oxygen molecule is small molecule and also non-polar. It can diffuse through the membrane. So that is how the bacterial cells get their oxygen. It diffuses directly through the membrane. And even in human cell, we use oxygen in the same way. In our lung, the oxygen will diffuse through the membrane into the bloodstream, into red blood cells. so that red blood cells can pick up the oxygen molecule. Okay, this is the cell membrane. And the function of cell membrane, first, they are selective permeability. And some molecule can pass through them, some cannot. Well, they also carry proteins on the membrane. Okay, a lot of membrane proteins. Some of the membrane proteins act as like facilitated carriers, facilitated channel, so that some molecule can pass through the channel. Or these molecules can be a transport protein. They can transport molecules from inside to outside or from outside to inside actively, but that costs energy by hydrolyzing ATP molecules. And also, the membrane is a site of electron transport and also oxidative phosphorylation, or we say that respiration of bacteria. So that on the cell membrane, it can produce a large amount of ATP. All the ATP molecules are produced on the membrane. And it can also excrete or enzymes from inside to outside. When it makes some enzyme, it can secrete the molecules from inside to outside, so that in the outside environment, the enzyme can break down big molecules for utilization by the bacteria. And the membrane also bury the enzymes and carrier molecules that function in the biosynthesis of DNA, cell wall polymers, and etc. It contains transport proteins that allow the uptake of metabolites and release of substances. So the membrane, even though it's a fluid-like structure, it can flow, and you can see that it can carry out important functions, important activities on it. And without the membrane, the cell cannot survive. But without the cell wall, it can survive, okay? The next part is the cytoplasm. Cytoplasm, composed by the abundant water inside the cell, contains a lot of water, and also protein molecules, nucleic acid, and also some lipid proteins. And it also contains a small amount of sugars and salts, like sodium, potassium, calcium, magnesium salts inside the cell. It carries the unique organelle of bacteria, that ribosome. Ribosomes are the only organelle in bacteria, but it's not a membrane-containing organelle. It contains only protein and RNA in it. And also, you can find plasmids in the cytoplasm and inclusion bodies. Ribosome? Okay. And we know in high school we learned that ribosome is what's the function of ribosome? Protein synthesis, right? Ribosome is like a tiny robot. It's like a machine. It contains two subunits, a big subunit and a small subunit. Altogether, it's a 70S subunit. It contains protein molecules and also ribosomal RNA molecules in it. It's a complex structure. It's like machinery, which can read the information in messenger RNA so that it can translate the information in messenger RNA into amino acid sequence to make polypeptides. So that's the only organelle of bacteria. And also plasmid. Plasmids are, well, relatively small DNA inside the cell. Usually they are circular DNA, covalent linked circular DNA. Smaller means, well, compared to the bacterial chromosome here, The chromosome is big, usually like million base pairs, several million base pairs. But for plasmids, it can be very small. Some plasmids can be like a couple of kilo base pair, a couple of thousand base pair. So it's much smaller than the chromosome DNA. And the plasmid, usually they carry some specific gene to give the bacteria some properties, such as antibiotic resistance, or in some pathogen, they carry the virulence gene, so that the bacteria carrying the plasmid can cause disease. Without the plasmid, they don't cause disease, because the virulence gene is on the plasmid. And plasmids are not essential for the cellular survival, which means that in the bacteria, if it carries some plasmid, well, the plasmid might contribute to some property to the cell. But without the plasmid, the cell can still survive, can still grow and replicate. Okay? So it's not an essential structure. This is an example of plasmid, the map of the plasmid, and you can see the size of this one is 4.36 kb. So that means 4,360 base pairs. Okay, inclusions of bacteria, we sometimes call them inclusion bodies. So that's something inside the cell. Sometimes when you stain the cell with some dye, you can see deep staining structure inside the cell, like this one. Okay, this is diphtheria. Okay, and this part. These are not the spore structure, but inclusion bodies. This contains a lot of phosphate groups in it, and the bacteria can utilize the inclusion body as the energy storage, so that when necessary, the cell can use this inclusion body to get extra energy. So that is why inclusions accumulate when a cell is grown in the presence of excess nutrients. So when the surrounding nutrients is very rich, so that the bacteria will make some inclusion body inside the cell. So when the condition becomes worse, like limited nutrients, so that they can use the storage for their survival. Okay, the next part is the nucleus. Well, we know in bacteria they don't have real nucleus. They have a structure called nucleoid. So which means that it does not have a membrane to surround the DNA, but the DNA is condensed in part of the cell, inside the cell. Here, the DNA is stained in red color. See? The region of this DNA dense structure we call nucleoid. And they don't have a membrane surrounding them. So that's a common property of prokaryotic cells. And in this part, that's the DNA. And we should understand that in a bacterial cell, the DNA must be well organized, so that they are well organized and packed. Because when a cell needs some gene to be expressed, you want to activate some part of the DNA, they can freely regulate that part and express that gene. And if the DNA is released from inside the cell, you can see this one. The actual length of DNA is much, much longer than the cell itself. See, this is E. coli. When DNA is released, you can see in each single cell, it contains a lot of DNA. It's very long. And you have to pack them tightly in this specific region. And this packing must be well organized. It cannot be like you have a pile of thread you just put into the box. It's not like that. It must be well organized. And when the cell needs some part of the DNA to be expressed, it can express that part. Okay, so even the bacterial cell is very simple, very small. It's still a very complex structure. And we have a lot of things to study on this single cell. Okay. Okay, the next we'll introduce the specific or particular structures of bacteria, such as capsules, flagella, pili, and spores. The first one I want to tell you what the capsules are. It's not in microbiology. When you heard the term capsule, what's the meaning of capsule? It's like that, the continuum medicine, that capsule. It's like that. So the capsule in bacteria, it's some extracellular polymer surrounding the cell. You have a cell like this, and you have some extra layer of polymer surrounding this cell. So it's like a capsule. And the material of capsule usually is polypeptide or polysaccharide. Okay? It can be polypeptide or polysaccharide. It depends on the type of bacteria. And if this material is condensed and with well-defined structure and layer, and this one we call it capsule. It's condensed with a little bit thick distance. This one we call it capsule. But sometimes the capsule is very, very thin. You cannot even see it with a microscope. And it's loose, not condensed, it's loose. And you can easily wash them away by like vortexing and centrifuge and get rid of them. And this structure, we call them slime layer. You know the meaning of slime, right? The sticky structure, slime layer. And the slime layer can be detached from the cell and remove them easily. For a lot of bacteria, they have a slime layer, and you can not actually see the structure, but you know there's some layer of material surrounding the cell because it can cover the antigen on the cell. And the components of capsule, it can be polysaccharide or it can be polypeptides. It depends on the type of bacteria. And like in this one of the important the capsule components of polysaccharide, like this one, okay, the hyaluronic acid, hyaluronic acid. For girls, you might know the name, right? Yeah, it's in your lotion. If you have it in your bag, you can take it out. Like in your lotion, it contains this one, hyaluronic acid. It's a polysaccharide, and because it contains a lot of this group, it can bind to a lot of water molecules, so that it can lock water molecules with them, so it can provide moisture on your skin. So that's the hyaluronic acid. So we can culture bacteria and purify this material and add it into our skincare. And in China, there are some companies fermenting bacteria to produce hyaluronic acid. So that is why now the price of this material is getting much, much lower than before. Because a long time ago, you have to isolate the acid from the animal tissue. And nowadays, you can produce them with bacteria. Okay. In my lab, we actually have this one. And I used to make some lotion with it. Not for me. It's useful, and you can add something to the lotion so that it can give you some moisture and also keep your skin like bright. So it's not difficult to make it. So that's a capsule. And when we study the structure of capsule, we can use special staining methods to see the structure. And like sometimes you can use a negative stain. A negative stain means that you are not staining the cell itself. You stain the background with some color, like this one. It's some ink. You stain the background with like black color, and the capsule part, it takes up the space, but it cannot be stained with black color. So that here you can see the presence of capsule. And also this one. So this is the presence of capsule. The function of capsule, and some of you guys are from BDS major, right? BDS. And this is a famous picture of biofilm on human teeth. The yellow color here, that's biofilm on human teeth surface. Biofilm is a special layer of bacteria. It contains a lot of bacteria in it. It attaches to the tooth, and it can grow a little bit thick so that it shows some color. And that's the function of capsule. The capsule helps the cell to adhere or attach to the surface. Okay? So that's a function of the capsule. And also the capsule protects the cell from phagocytosis. You know phagocytosis, right? When we learn immunology, we learn that. And yeah, if a cell without a capsule, the antibody can recognize the antigen on the surface, and then through the Fc receptor, okay, the Fc receptor binds to the antibody, okay, it can initiate phagocytosis to get inside the cell, okay? Then finally, the bacteria is digested and killed, fused with lysosome, okay? Then the bacteria is killed and degraded. But if the bacteria has a capsule layer like this one, the antigen is already covered in the capsule so that the receptor in the immune cell cannot recognize the antigen, the ligand, so that it protects the cell from phagocytosis. It's safer for the bacteria. And also, the capsule acts as a barrier to antimicrobial substances in either the blood or in the body fluids. And those molecules, these antimicrobial molecules, include antibiotics, complements, and lysozyme. So it's a physical barrier surrounding the cell so that it can provide some protection. Okay, the next structure we want to introduce is called flagella. Flagella is a thread-like structure, and they are the organ of locomotion and can provide motility for bacteria. And all the bacterial flagella are composed of protein molecules, and those protein molecules, we call them flagellins. Okay, that's a protein name, flagellins. They are actually the H antigen of bacteria. When we say the H antigen, you know that bacteria should have flagella. So the H antigen is the antigen on the flagella. The flagella can be much longer than the bacteria itself. Well, the bacterial cell, we we talk about that, it can be like a couple of micrometer long, but the flagella can be, well, 10 to 20 micrometer long. So that's much longer than the cell itself. But it's not very thick. The diameter is like nanometer, 12 to 30 nanometer. If the flagella is stand properly, you can actually see the presence of flagella with a light microscope. But you have to use a special method to stain the flagella so that this flagella might look much thicker than it should be, so that you can observe it with a light microscope. Okay, so this is the electron microscope showing us the flagella. Some flagella, there is only one flagellum on one end of the bacteria. This one we call the monotrichus. Mono means what? One. So one flagella. And this one is called amphitrichus. So on each pole, there is one flagellum. So this word amphi means two, right? Two flagella. While this one, it has a lot of flagella surrounding the cell. So this one we call it peritrichus. Peri means surrounding. And this one, there is a bunch of flagella on one end of the cell. Okay? I call it luphotrichus. Okay? Different type of flagella. Let me show you how the flagella works. The flagella actually can be found in both gram-positive and gram-negative bacteria. This is gram-negative, this is gram-positive. And one end of flagella is actually anchored to the cell membrane and cell wall. The cell membrane and the cell wall, okay? It attached to it. And it contains a lot of protein molecules to form a well-controlled machinery on the membrane. Let me show you the machinery on molecular level. It's well designed. It's like a made in a factory. This is the actual flagella on the membrane. It has this, okay, ring-like structure, all made up by protein molecules. And it has a motor molecule like this, okay? And when it catalyzes the hydrolysis of ATP molecule to release energy from ATP molecule so that the ring can rotate. And when the ring rotates, it can carry the flagella to rotate. And rotating of flagella like this can, well, push the bacteria to either rotate or move or swim. Okay? So that's how flagella works. A decade ago, we only know the structure like this. Okay? And nowadays, with the help of new techniques such as the cryo electron microscope, we can study the membrane protein on molecular level, on like even atom level, so that we can finally decipher how they work. It's a well-designed structure like this. This is the function of flagella. It's the organ of locomotion, so they can provide motility for bacteria, allowing the cell to swim toward food and away from poisons, so that they can sense the presence of gradient of nutrients, so they can swim to the nutrients. And if there is some poison or some toxic molecule, they can swim away. Okay, the next structure is pili. Pili is much shorter than flagella and much thinner. It covers the surface of the cell, this hair-like structure. They are rigid surface structure. and which are shorter and finer than flagella. And pili are composed of proteins called pilins. Flagella is composed by proteins called flagellins. Okay, pilins. And there are actually several different types of pili. And we'll introduce two types. One is the ordinary pili, or the common pili, ordinary pili. They are short, like this one, short and straight hair-like structure. Another one is called sex pili. Sex pili is much longer and flexible, so that the bacteria can use sex pili to exchange DNA. This is ordinary pili. Okay, these structures. They play a role in adherence. The pili can adhere to the human tissue or human cell. They attach to the surface of human cell. And they are related to virulence of pathogenic bacteria. With the pili, they attach to the tissue surface. They can colonize and cause infection. But without the pili, they can be washed away. And the sex pili is a little bit longer, and it's a hollow pili and more flexible. It's this one. Okay? The function of sex pili is for conjugation or genetic material exchange. For example, if this bacteria has a sex pili, it can actually grab another bacteria. Then the pili will get shorter and shorter so that it can draw the two cells together, like this one. Bring the two cells together. When the two cells attach to each other, there will be a channel formed on the interface. Okay, and through the channel, the donor cell, this one, the donor cell will copy their DNA or a plasmid DNA into the recipient cell, and finally, both the donor cell and the recipient cell will carry the same plasmid. So this procedure we call conjugation. Okay. And through conjugation, the donor cell is able to transfer their plasmid or even genomic DNA into the recipient cell. So that's a useful tool, right? In the lab, we can use conjugation through the sex pili to mediate the DNA transfer. The last structure we want to introduce is the endospores. Endospores are not found in the replicating bacteria. Only in special conditions, a number of gram-positive bacteria can form a special dormant structure inside the cell. For example, when the nutrition is getting limited, and this special structure we call endospore. And the other property is that one cell can form only one endospore. So that the endospore is not a real spore like in fungi. It's only a dormant form of the cell. So they just become dormant or rest for a long time. But one cell can form only one endospore. When the condition is becoming better, becoming like a favorable condition, for example, when the nutrients is getting better, and the endospore can germinate. Okay, germinate means that they can form replicating cells. They produce a single vegetative cell. One spore can only form one vegetative cell, and this vegetative cell is a growing replicating cell. And yeah, the spores are commonly found in general like Bacillus and Clostridium. Bacillus and Clostridium. And in future, we'll introduce some important pathogen in this genus, Clostridium bacteria. And in our lab, currently we are more interested in Bacillus genus because in this genus, some of them can produce antimicrobial molecules. Maybe antimicrobial peptides or other molecules. And probably some of them are useful to make new antimicrobial medicine. This is the example of endospore, okay? The structure like this. So that it has several layers of different material inside the endospore, and the DNA is in the core part, so that the cell will become dormant for a long time. And the characteristic property of this endospore is that they are very resistant to heat and to other factors. Like in the boiling water, you can kill vegetative bacterial cell in several minutes, but for endospore, they can survive in boiling water for like half an hour or 1 hour, so that you need to treat the bacterial endospore for a longer time to kill them. Or in the lab, when we use the autoclave, we can increase the temperature to like 121 degrees, and the endospore can survive for like 15-20 minutes. So they are more resistant to heat. This is endospore. And the endospore, it can be formed in different parts of the cell. Some form in the center part, so this is central endospore. Some form in the exact terminus, so we call it terminal endospore. And this one is close to the terminus, we call it subterminal endospore. And some endospore diameter is bigger than the cell, like this one. So it will form this swollen end endospore, drumstick-like endospore, so that you can find characteristic structure like this, and it can help you to determine the type of bacteria. Okay, this is endospore. They're highly resistant to environmental factors such as desiccation, heat, and chemical agents. And endospore can exist for a very long time, even decades or hundreds of years. And then when the condition is better, they can germinate again. And usually, the endospore itself does not cause disease, but only when they germinate to form vegetative cells, they can cause infection and disease. Okay? And it's hard to kill them, so we usually treat endospore at 121 degrees for 15 minutes or longer to kill them. And that's the reason why we can use endospore to make sure the autoclave is working well. So in the hospital, you regularly put endospore kit into the autoclave, treat it, then culture it. If you cannot culture the bacteria, it means that all the endospores are killed. So that means your autoclave is working properly. So it's okay. But after treating the endospore, if the endospore can germinate to grow again, which means that your machine needs to be checked and repaired. So we can use that as an indicator. Okay, there is a short video. We don't have time, I think, right? Okay, that's all for today.